This module of Chapter 10, Module 10.5, is on the use of the multiple decrement life table in biological control of insects. Biological control is a method of insect and plant pest control based on the use of natural enemies, including predators, parasitoids, and pathogens. This may involve either introducing natural enemies from world regions where the pest is endemic, using mass-released predators or parasitoids, or biopesticides such as the microbial pesticide Bacillus thuringiensis, or by conservation methods aimed at increasing populations of naturally occurring predators and parasitoids. Measuring the impact of natural enemies on pest survival and population growth is a central component of studies in biological control. It thus follows that tools are needed to quantify the effect of natural enemies on pest populations. In 1989, I introduced the multiple decrement life table to the ecology literature as an alternative to key factor analysis. And later, Robert Peterson and his colleagues built on and applied the multiple decrement concept in insect pest control with particular emphasis on irreplaceable mortality. The concept of irreplaceable mortality was first introduced in a study concerned with the impact of two parasitoids on the biological control of a pest scale insect, where irreplaceable mortality was defined as that portion of the parasitism which, if it were lacking, would not be replaced by some other cause of death. Here I define irreplaceable mortality with reference to the competing risk and multiple decrement life table concepts I introduced in Chapter 8 as the marginal difference between all-cause mortality and mortality due to the elimination of one or more risk factors. In a hypothetical example, consider mortality and survival through the three stages of egg, larvae, and pupae. All stage mortality is 0.1, 0.4, and 0.9 for these respective stages, yielding a survivorship of 1,000 as a radix, and then 900, 540, and 54 individuals reaching adulthood. If we eliminate egg mortality, then survivorship of 1,000, 1,660 reach adulthood. Therefore, irreplaceable deaths equal 6. The explanation is that 94 out of the 100 that originally died in the egg stage died instead in either the larval or pupal stage, leaving 6 extra survivors. This outcome can be contrasted with the outcome when larval mortality is eliminated, in which case irreplaceable deaths equal 36. The explanation here is that the 324 out of the 360 individuals that originally died in the larval stage died instead in the pupal stage, leaving 36 extra survivors. To illustrate this concept more concretely, I present a real example from the field study of the alfalfa weevil by Robert Peterson and his colleagues, where they quantified multiple sources of mortality for this pest and brought multiple decrement theory to bear on analysis of the problem. Here's a table from their study in which this first column contains different stages of the insect. The next column gives the stage-specific mortality. The next cohort survival in the following column, LX, and the last column, the fraction of the cohort that died in each stage interval, which is DX. Note that the total deaths equal 0.926 of the original cohort, and at the top of the next 5 columns, this fraction is disaggregated by cause, including due to wasp parasitism, infertility, failure to develop, fungal pathogen, and rain. The sum total of these equals the total deaths of 0.926. This cause of death information in the presence of all causes is used to estimate probabilities of cause of death in the absence of other causes, including for the wasp, infertility, failure to develop, fungal pathogens, and rain. This information in turn is used to compute irreplaceable mortality as shown in this table, with columns of various combinations of causes, the percent mortality due to this combination, the causes that are eliminated, and the percent irreplaceable mortality due to these eliminated causes. The most important outcome of this analysis is that of identifying the important role of a fungal pathogen in the overall dynamics of this particular population of alfalfa weevils. Note that for fungus alone, nearly 60% of the total mortality from all other causes would not be replaced. The importance of this factor is underscored when elimination of this factor is combined with the elimination of other factors, showing the minimal impact of the others when combined with a fungus as a cause. The one exception is elimination of both failure to develop and fungus results in over 80% of the deaths that would not be replaced. I will end this module by noting that identification and quantification of causes of death in natural populations is hugely challenging, both in theory and in practice. Indeed, accurate databases on causes of death in the wild are exceedingly scarce for any non-human or non-domestic species, and represents an area of ecology and biodemography in dire need of more research.